Hi, welcome to yet another session of biology classes with Lakshmi Chandra. As you all know that we were dealing with the structure of nucleic acid and we have completed the structure of DNA, and I have promised you that I will be coming in the next session with the different types of DNA. So this session is devoted for that. That is the different types of DNA. Okay. So let's look at that. Types of DNA. It's actually exciting to know, isn't it? DNA, that is our genetic material, occurs in different forms. Okay. We might be thinking that there is only one form of DNA, but it's not so. The forms of DNA, that means the structure of DNA that we have learned in the last session, it differs according to the environment in which it is put. So let us first see that. Okay, the different forms of DNA and the different environments in which they are put. The different environments means we have to specify the amount of water or the relative humidity present and the ion concentration. So the forms of DNA change according to the relative humidity and the ions present over there. So let's first focus into that particular parameter. So depending upon the presence of humidity, the amount of humidity and the ions present, you have 4 different types of DNA or 4 different forms of DNA. It is designated by the alphabets: DNA A, DNA B, DNA C and DNA Z. So I told you the environment, isn't it? That means the amount of water or the relative humidity and the ion concentration. Among these 4 DNA types, it is the bDNA. It is the bDNA which is physiologically occurring form of DNA. That means normally occurring form of DNA is bDNA. The Watson and Crick model of DNA is actually the bDNA. So when does that occur? It occurs when the amount of water is very high or the relative humidity is actually around above 90% or it is around 92%. When the amount of humidity is 92% and when the amount of ions is relatively low, that is the normal physiological condition, the form of DNA that exists is B form of DNA. But then what happens? When the amount of water is reduced to 75% or so, that means the humidity is reduced, the form of DNA that is seen at that time is called as the A form of DNA. So in this A form of DNA, the properties, that means we have learned about many properties of DNA, not actually the features of DNA we have learned, isn't it? We have learned about the major groove, the minor groove, the pitch, the helix, the rays of the helix, isn't it? Distance between the distance within a turn, distance between 2 base pairs. So many concepts we learned in the previous session. It will be better you can refer the previous session and then look into this particular session so that you will get a clear understanding of what the structure of actual DNA is. And after that, if you look into this one, you will clearly understand the differences. Okay, so coming back, the A form of DNA is seen when the amount of water is reduced to 75% and when these ions are present. That means sodium, potassium or cesium ions are present. At that time, the bDNA takes another form. That is, the DNA occurring at that particular form is A form of DNA. But then what happens if still the humidity is reduced? That means the humidity, relative humidity is brought to 66% and if the amount of lithium ion is very high, okay, the amount of lithium ion is very high, then the form of DNA that occurs is C form of DNA. Now when the relative humidity is extremely low, that means the amount of water is very less and the amount of ion concentration is very high. At that particular environmental condition, the amount of DNA or the form of DNA that occurs is called as the Z form of DNA. So what I have just told you is that depending upon the environment, that means depending upon the amount of ions present, the nature of ions present, the amount of water present, the forms of DNA can be different. Okay, now let's look into the salient features of each form of DNA. Let's look into one by one the salient features of each form of DNA. First, you have to learn about the helical turn. Except Z form of DNA, the rest of the three forms are a right-handed helix. Now, what is that right-handed turn? What is that right-handed helix? Okay, imagine there is a thread and it is going to turn now, and first turn it is turning towards the right side and then moving up like this. That is called as the right-handed helix. See, I'll show you like this. Right-handed helix is like this. The DNA is moving towards the right side first twist, and then it is moving like this. Okay. Now the other strand starts from above. Again, it is first turning towards the right hand side, and then it is moving like this. So this is the right-handed turn, right-handed form of DNA, or you say the helix is having a right-handed turn. That means it is having from here, this is my right side, so it is moving towards the right side. Like this the movement is. So that is right-handed helix. Now I told you A, B and C DNA forms, they are having right-handed helix. But then what is that left-handed helix? In left-handed helix, the first turn should be towards the left side. Okay, so it is like this. From here, the DNA turning is towards the left side, and then like this, like this, like this it goes. Okay, then again from there, from top, opposite side. That means again from the left side only it goes. So it's like this. Like this. So that is left-handed form of the helix. That is left turn. So here the beginning is from this point it moves towards the left side. So that is the right-handed helix and the left-handed helix. Okay. So I hope the first point is clear to you. That means the helical turn. First point was regarding the environment. I told you when it occurs in A, B, C or Z form. Second point is regarding the turn of the helix, the pattern of turn, whether it is right-handed or left-handed. I hope now it is clear to you with regard to what is right-handed and what is left-handed from the diagram. Okay. Now next third point is the diameter of the helix or the width of the helix or the distance between the two strands. Okay, the diameter or width or the distance between the two strands, what is that? In the case of B DNA, we all know it is 2 nanometer. Last session I explained to you this width of DNA. That means the distance between these two strands. This is the width of DNA, or this is the width. Here, this is the width. Okay, so when you look at this particular forms of DNA, A, B, C and Z form of DNA, in the case of Z form of DNA, the width is extremely less. Extremely less. Okay, the distance between the two strands is extremely less. It is just 1.8 nanometer. 1.8 nanometer. But in the case of C, it is a bit higher. 1.9 nanometer. Then in B form, it is 2 nanometer. And in A form, it is 2.6 nanometer. So that is with regard to the width of the helix or the diameter of the helix. I hope that is clear to you. That point is clear. Now next one, coming to the helical pitch. So what is this helical pitch? That is distance of one turn. So actually what is the distance of one turn? Actually these points were dealt with in the previous session in detail. Anyway, I'll just tell you what is that pitch of the helix. See, if you're drawing a DNA molecule like this, DNA molecule is drawn like this. Okay, here you can see the distance or the width is too large. Okay, here it is less, and here again it is too deep and large. So this area where the distance, the diameter is very high, you call that area as the major groove. Major groove. Okay. Here you can see this is the major groove. But here you can see this is a minor groove, isn't it? The width is very less, minor groove, or the depth is very less. So that is called as a minor groove. If the depth is too high, okay, you call that as a major groove. Major groove. Understood? So here, the distance, this is a major groove, this is another major groove. So the distance between these two major grooves, that you call, that is taken as one turn, or that is called as the pitch of the helix. Pitch of the helix. Distance between two adjacent major grooves, or the distance between two adjacent minor grooves is called as the pitch of the helix. So that pitch of the helix, that also is having a number. In the case of bDNA, it is 3.4 nanometer, or the distance of one turn of DNA helix. That distance is 3.4 nanometer. And in that one turn, how many bases are there? There are 10 bases. In that one turn, there are 10 bases. What about our aDNA? The distance of that one turn is 2.8 nanometer. In the case of cDNA, it is 2.9 nanometer. And you see the case of zDNA, the distance between of that one turn is too large. It is 4.5 nanometer. And actually, when you draw this zDNA, the zDNA, from the name itself, it is having a zigzag appearance. It is having a zigzag appearance because of this property. This turning is towards the left hand side, it happens. The width is too less, okay? But the distance between the two grooves is very high. So it is having a zigzag appearance. That's why the name itself is like zDNA. So distance between two major grooves or minor grooves, or the pitch of the helix, that is 4.5 nanometer. Okay, and in that 4.9 nanometer, how many bases are there? There are 12 base pairs. So that is with regard to number of base pairs per turn. In that one turn of that zDNA, you are having 12 base pairs arranged one after the other. But coming to cDNA, it is just 9. In the case of bDNA, it is 10. This we all know. And in the case of aDNA, it is 11. So if you examine the zDNA, it is formed when the humidity is extremely low and the ion concentration is extremely high. It is having a zigzag appearance. It is a left-handed form. Turn is left-handed. Okay, and width is too less, 1.8 nanometer only, but the distance of one turn is very large, that is 4.5 nanometer, and in that one turn there are 12 base pairs. Coming to bDNA, that is our normal DNA, the distance of one turn is just 2 nanometer, and in that, the distance of one turn is 3.4 nanometer, and in that 10 base pairs are arranged. So, what is the distance between two adjacent base pairs? That is, in one turn there are 10 base pairs and the total distance is 3.4 nanometer. Okay, so what is the distance between two adjacent base pairs? It will be the total distance divided by the number of base pairs. That is 3.4 divided by 10. So 3.4 divided by 10, that is 0.34 nanometer. What is that? That is the distance between two adjacent base pairs. Together two base pairs are there. The distance between two adjacent base pairs, one after the other, base pairs are coming, isn't it? Distance between these adjacent base pairs is 3.4 nanometer. Sorry, 0.34 nanometer. How do you get that? You divide the total length divided by the total number of bases present in that particular length. So that is 3.4 divided by 10. So that is 0.34 nanometer. Same thing we can apply here also. In the case of aDNA, the distance, the pitch of the helix is 2.8. Pitch divided by the number of bases per turn, that is 2.8 divided by 11, you will get 0.25 nanometer. So 0.25 nanometer is the distance between two adjacent bases in the case of aDNA. And in the case of cDNA, if you divide these two, you will get the value 0.33. Same is the case with zDNA also. 4.5 divided by 12, you will get 0.37 nanometer. So this particular column says about the distance between two adjacent base pairs. So that distance between two adjacent base pairs is given the name as the helical race. Helical race. Okay, helical race. So with respect to the helix, we have to learn about what is the nature of the turn. Except zDNA, all others are right-handed. What is the diameter of the helix? The width. Okay. Then we have to learn about the distance of one turn, that is the pitch of the helix. Then how many bases are there in one turn, that is we have learned about. In the case of bDNA it is 10, minus 1 is cDNA, plus 1 is aDNA. But in the case of zDNA it is 12. Okay. Then we learned about helical race, that is distance between adjacent base pairs. If you just divide the pitch by the number of bases present, you will get the helical race. Then we have to learn about what is the nature of the major groove and the minor groove. The major groove, except in the case of aDNA, the major grooves are wide. Okay, the major grooves are very much wide and they are very much deep, except for aDNA. In the case of aDNA, the major group is narrow, the width is narrower. But in the case of zigzag, that is Z form of DNA, the major group will be completely flat like structure. You cannot see the turning properly of the DNA. It will be a flat structure. Okay, so that's why it is written as flat here. So major groups are wide in the case of B and cDNA. It is flat in the case of zDNA, but very narrow in the case of aDNA. Now look at the minor groove. Minor groove is just opposite. aDNA will be very wide. The minor groove will be very wide and it will be very deep. Okay. But the other three forms of DNA, the minor grooves are very narrow. So these are the features of A, B, C and Z DNA. When you write for the exam the different forms of DNA, first point you have to write is why these different forms. It is all because of the environment in which it is present. Environment means you have to specify the amount of water and the amount of ions present, the nature of ions present. So accordingly the nature changes. A, B, C and Z form of DNA you will get. Okay. And each of these DNA will have a different numerical values with respect to their pitch, with respect to their diameter, the number of bases, the nature of the major groove and the minor groove. It's not over. Actually, we have another form of DNA also called as the D form of DNA, ddNA. Okay, that is a mysterious DNA. It is a peculiar type of DNA. So that also I'll explain to you. So ddNA is there. Plus you should also know about a kind of DNA that is present in prokaryotes. What is its nature? And there is another cDNA also. Here I told you cDNA. Another cDNA is also there, children. When you write this C as small letter C, DNA, what is that? So these are some exciting things. So that also will be dealt with in this particular session itself. Having completed the different forms of DNA, that is A, B, C and Z forms of DNA, let's move on to some very rare forms and some different forms of DNA. Okay. First let us look into this form, D form of DNA. This form of DNA is an extremely rare form of DNA. And the peculiarity is that in this DNA, the guanine residues are absent. That means the D form of DNA is devoid of guanine residues and it is an extremely rare form of DNA. More have to be found out regarding this D form of DNA. Some of you can also take up these research activities and come out with success, okay, regarding this D form of DNA, its importance and all. I hope some of my students do research on this and come up with success. So D form of DNA is an extremely rare form of DNA in which the guanine residues are absent and there are only 8 base pairs per turn. In that one turn there are only 8 base pairs. Okay. Now having completed those alphabetical nature, that is A, B, C, D form and Z form of DNA, I also told you that when you write a small letter C and then DNA, it is different from the capital letter C DNA form. So what is that small c DNA form? or see, it is written like this, cDNA. Here, the small letter C represents the complementary DNA. So what is that complementary DNA? And how does it differ from the normal DNA? Let us look into that. See, in the case of normal DNA, suppose you have the bases adenine, thymine, guanine, cytosine. Again, I am writing here 2 thymine molecules. So here, this is the bases that are present in one strand of DNA. Okay. When this particular DNA produces an RNA, or this gives information for the production of RNA, the RNA molecule formed will be complementary to this strand. So here, this is the DNA strand, and this DNA is now giving information for the production of RNA molecule. So how the RNA molecule will be? The bases in the RNA molecule will be complementary to the bases in DNA. So adenine pairs with thymine. So I am writing here T, then A, C, G, A. How did I write this? Complementary to the bases present in DNA. What is a complementary? It's the base pairing rule, the Chargaff's rule that we have learnt in the previous session of the DNA structure. But here what I have written, the next strand is my RNA, isn't it? But I told you in the previous session the difference between DNA and RNA. In RNA, thymine is not present. Instead of thymine, what is present? Uracil is present. So when I write RNA, I cannot write thymine there, isn't it? Though adenine pairs with thymine, since I am writing RNA, I cannot write thymine, but I have to replace the thymine with uracil. So I am replacing thymine with uracil. So DNA has given rise to RNA. Now in this point, let me tell you one more thing. See, imagine we are having 10 base pairs of DNA now in one cell. There are 10 base pairs, you imagine. All these 10 base pairs will not be functional. Okay, among these 10 base pairs, there will be only 5 or 6 base pairs which are actually functional. Okay, this point you have to keep it in mind. Imagine there are 10 base pairs. It is not 10 base pairs. There are 3.6 billion base pairs are there. Okay, imagine there are 10 base pairs, and out of these 10 base pairs, actually only very few, that is only 2% will be actually functional. Those functional regions of the DNA, okay, which are actually functional, once this DNA is producing this RNA, and among this, so imagine only these 3 pairs are functional. Okay, so only these 3 pairs will be retained in this RNA. Rest of the bases will be removed. A detailed mechanism of how this is retained and how the other things are getting removed are coming in the coming sessions. Do wait for that. That is named under a process called as transcription. So now for you to understand what is cDNA, small cDNA, this complementary DNA, I am telling this in nutshell, in short. Okay, so we have DNA that is giving rise to RNA, and among that the whole thing, the whole bases are not functional. Some of them are not functional, so I have removed them. Okay, now again, after removing the non-functional parts from that RNA which is produced, okay, I have removed that, and from there another DNA is getting formed. So that will be complementary to this RNA, isn't it? So it will be thymine, guanine, and guanine is going to pair with cytosine. So this is the actual functional DNA. Actual functional DNA. So what is that actually? This was a normal DNA present in the cell that had produced the RNA according to the complementary rule. From that, the non-functional regions are all removed, and from there again another DNA is getting synthesized, and in that DNA which is getting synthesized, all the base pairs will be functional. So this DNA which is produced from the RNA and in which all the base pairs are functional, you call this DNA as the small c DNA or the complementary DNA. Okay, this particular part, the complementary DNA or the cDNA, is of great importance in the field of biotechnology. That also will be dealt in detail in the coming sessions. Do wait for that. Okay, so cDNA, here since I am dealing with the different forms of DNA, I just told you that there is another form of DNA called as complementary DNA. It is a totally functional DNA. There will not be any region in that DNA which is not functional. So that is the difference between the cDNA and the normal DNA. So this cDNA is actually obtained from RNA. Okay. Now moving on to the next form of DNA, that is circular DNA. Circular DNAs can be single-stranded or double-stranded. Usually, in the case of prokaryotes, there is a region called as nucleolus where its genetic material is present. So the genetic material of prokaryotes are circular DNA or single-stranded circular DNA molecules are present in the case of prokaryotes, like in bacterium and all. Okay, and it is present in a region called as nucleolus. Not only there in prokaryotes, in eukaryotes also we can find this circular DNA, single-stranded circular DNA. And in which part of the cell? It is actually present in two organelles called mitochondria and plastids or chloroplast. In these two organelles, inside the eukaryotic cell itself, they are having single-stranded circular DNA. But otherwise the eukaryotes in their nucleus, they all have a linear DNA. So the part of the eukaryotic cell where you can find circular DNA are the two particular organelles, that is mitochondria and chloroplast. And they resemble the prokaryotic nature in this case. Okay, prokaryotes also have single-stranded circular DNA and that is present in the nucleolus. Now look at this one. Double-stranded circular DNA called as plasmids. It's very important. It's have got a lot of applications in the field of biotechnology again. Plasmids. They are double-stranded circular DNA and they are found mostly in prokaryotes. Double-stranded circular DNA. Imagine this is the bacterial cell. Okay, bacterial cell, and you have the circular DNA. I'm just drawing it like this. Okay, circular DNA here in the nucleolus. Apart from this, some of the prokaryotes, not all, some of the prokaryotes, they have an additional double-stranded circular DNA. Double-stranded circular DNA. Okay, and this is found outside its normal chromosome, isn't it? So you call it as this is extra chromosomal. You call it as extra chromosomal because it is found outside the usual nucleolus region. It is found outside. It is not found along with that. It is found elsewhere, but inside the cell. Okay, so extra chromosomal, double-stranded. It is double-stranded, and how is it? Circular. Circular DNA molecule, and it is capable of self-replication, that is independent replication. Replication means multiplication. That means it is not depending on this chromosomal DNA in any way for it to multiply. It is having its own mechanism of multiplication. So it is self-multiplying. Self-multiplying. Okay, or self-replicating. Self-replicating, extra-chromosomal, double-stranded, circular DNA molecules are present in some of the prokaryotic cell. And you know what it does? It imparts some additional properties for the cell. Some additional properties for this particular cell, like antibiotic resistance and all. Imagine you have the plasmid, this is a bacterial cell, and this bacterial cell is having a plasmid inside that. So when I say plasmid, this figure should come to your mind. How is it? It is outside the chromosome, extra chromosomal, double-stranded circular DNA. And what is the peculiarity? It is capable of independent replication, self-replication. Okay, and it is also imparting some additional properties. So I'll tell you what's that additional property. See, imagine this plasmid is present, and this plasmid is having a region here, this particular region of the plasmid. Okay, this region is imparting antibiotic resistance. For example, say the antibiotics penicillin, the first antibiotics found out, penicillin. This particular region is having a property to actually resist the effect of the antibiotics penicillin. That means if penicillin is added, it will kill this bacterial cell. But if this bacterial cell is having the plasmid to resist the effect of penicillin, what will happen? The bacterial cell can resist the effect of penicillin. That means the bacterial cell will not die. That means even in that penicillin medium, the cell can survive. So that is that kind of additional properties these particular plasmids they impart. And the biotechnologists have made use of these additional properties, the self-replicating properties, the circular small nature of this plasmid to actually frame many new biotechnological products. But all these things we'll be learning in the coming sessions. Okay, so here in today's session we have dealt with the different forms of DNA. So if we simply rewind, the DNA occurs in different forms according to the environment in which it is present. So accordingly there are A form, B, C and Z forms of DNA, and there is a D form of DNA which is extremely rare. There is small c, that is complementary DNA, which is having all the bases which are functional produced from the RNA. And then you have single-stranded as well as double-stranded circular DNA. The double-stranded circular DNA called as plasmids have got its own peculiarities and applications in the field of biotechnology. And the normal DNA that is present in our cell, that is nuclear, that is linear DNA. So I hope you have understood the different forms of DNA. Okay, and I humbly request you to actually refer the previous sessions of structure of nucleic acid, then structure of DNA, so that whatever I have taught you today will be understood in a better way. Thank you for now.